Hej och välkomna till Hamlet BioPharmas nya webbserie där vi ska introducera mer om hur vi jobbar och ni får träffa medarbetare med kompetens inom olika delar av läkemedelsutvecklingen. Ni ser bakom mig Ine som vitt och Arunima Chowdhury som är de som ska presentera sitt speciella arbete idag. Och labbet är också fullt av alla i gruppen som jobbar. Martin och Jakob är i bakgrunden, så alla är här. Och vi vill också ha en dialog med er som är öppen, full av frågor. Ni kan stanna, be den som pratar göra ett stopp och ställa konkreta frågor till olika bilder eller ta upp annat som ni är intresserade av att diskutera. Så det är en helt öppen diskussion, så som vi brukar ha. Jag ska nu be att få dela skärmen så att ni kan se det material vi har tagit fram för idag, och vi kommer också att byta från svenska till engelska eftersom vårt labb är en mycket internationell arbetsplats. Nu ska vi se här. Så som sagt, välkomna till Hamlet BioPharma och vår verkliga verkstad där mycket av arbetet pågår. Den här första bilden ger en översikt över läkemedelsutveckling som vi är uppfostrade att se, hur man gör och i vilken ordning saker händer. Först till vänster ser man upptäckten, vetenskaparna som upptäcker ett nytt koncept, i vår fall antal koncept, eftersom vi har många olika. Kan ni stänga av era mikrofoner? Tack. Så från upptäckten så ska man ta sig hela vägen fram till ett godkänt läkemedel. Och då känner ni alla till de här stegen. Det behövs vetenskap och teknologi till att börja med, alla molekylära verktyg för att identifiera och karaktärisera det nya läkemedlet. Det behövs djurmodeller för att veta om läkemedlet fungerar och har behandlingseffekter. Det krävs hela apparaten med translation, både för produktion av läkemedel, man ska träna sig på tox, man ska ha kompetens att bestämma hur läkemedlet ska ges och vilka som är i patientpopulationen som är lämplig. Och sen slutligen då den kliniska delen. Och det är klart att allt detta är sant, men i verkligheten så är det lite mer komplicerat. Och egentligen så är det så att alla de här kompetenserna behövs samtidigt. Om ni ser på Novo Nordisk som modell för läkemedelsutveckling, en fantastiska historia, så har man där lyckats att under hela utvecklingsperioden för läkemedlen också hålla en väldigt hög vetenskaplig kompetens. Så att man i varje steg i processen kan ha en faktabas för vad man väljer att göra, hur man producerar sitt läkemedel, vilken patientgrupp som ska behandlas, hur man ska utvärdera effekterna, hur marknaden ska se ut, vem som är en bra kommersiell partner och så vidare. Och det innebär att vi i vår lilla skala, vi försöker att kombinera alla de här kompetenserna samtidigt, och det som vi kallar för vår drug development platform. So we can look at the compound, our discovered compound, in great detail, almost artificial intelligence detail with technologies like NMR and crystallization. We have to know a lot about disease mechanisms and biology. As you will hear today, you can see details in the biology or disease process by high-resolution imaging. And especially, of course, cancer biology and infection biology are very, very, very important for us with the drugs we are developing. There is a lot of complex information here that one needs to have access to in order to do a good job. Genomics, transcriptomics, you will hear more about that in the upcoming web meetings. Animal models of disease, medical expertise, tissue evaluation technology, and on the business side, of course, legal, finance, patents, drug production technology, regulatory advice, clinical trials expertise. Actually, all of these competences need to be integrated. And in our case, of course, this is achieved because of the very special people who are here, many who are here today, and also our network of international, highly competent co-workers that help us further the process. So I will now leave the word to Aranina Chaudhary and Ines Ombit, who will tell you more about one aspect of the work, which is our imaging platform. So Aranina, please.
Good afternoon, everyone. It's a great privilege to share with you the excitements of our imaging platform. So we've used the state-of-the-art microscope, a confocal microscope, and a spectrum of different fluorescent tags to image cells in cancer, in infection, and in various clinical models with the purpose to understand disease and find a cure. It's all about cells, but most of you really don't know what cells are. So this is an animation which gives you a bird's eye view of how cells look like and how they are the building blocks of larger organized tissues and organs. So I will just play it again. So you can see that this is a very Barbie kind of an image of a cell, but it gives the animation gives a good realistic version of how this organizes into tissue. But in reality, a cell is a much more complicated structure. And on the right is an image that is more realistic to what it is. A cell is surrounded by a boundary, and it encloses a universe in itself, which contains various cellular compartments. And nucleus is at the heart of the cell, which contains all the genetic material, DNA, RNA. And the imaging technology allows us to basically look at each of these cellular components in great detail. To give you an example, on the left is a more realistic image of a cell where the different flags point out to the different organelles along with the function. So here on the right is shown a cellular organelle called endoplasmic reticulum. This organelle is shown in live cell, and we have used a green dye. That's why it looks green to allow us to visualize the endoplasmic reticulum. It is the assembly line that spans all the way from the cell membrane. As you can see, it nicely encloses, or you can see the periphery of the cell with this fine tubular network, and it encompasses all the way from the plasma membrane to the cell center. And in this high dense network is the nucleus, which is not shown. And as you can see that the microscope allows us to actually see the different parts of the endoplasmic reticulum. And therefore, when we add our drugs of interest, for example, alpha-1 oleate, you can immediately see the effects and you can evaluate what it is doing to the different structures. Now going to another important structure, it's the cytoskeleton of the cell. And you can say that these are these railroads, these fibers that give strength to the endoplasmic reticulum, keep the structure of the cell intact so that the cell can perform various functions. And again, the imaging allows you to see that how this dense network is assembled. And again, in the center, you can see the nucleus, and you can see the organization of these structures in great detail. Here is another pattern of the powerhouse of the cell, which is called the mitochondria. So the mitochondria, and this is again live cell, and we have used a green dye to visualize the mitochondria. It is the powerhouse of the cell because it generates energy that gives something called ATP for the cell to perform various functions. And the power of imaging allows us to see the remodeling, the structural changes that this very important organelle undergoes with various drug treatment. And this is another organelle, which also looks very different from the ones that I showed you. This is called the Golgi. It is the distribution center. And I have done 3D reconstructions here which allows you to understand or appreciate the topology of the association. In blue is the nucleus, and in brown is the Golgi around it. And Golgi is like the distribution center. So when the proteins are made by the endoplasmic reticulum near the nucleus, the Golgi actually sends it to the various locations where they perform sophisticated functions. So this gave you a flavor that the powerful imaging technology allows us to see these different cellular components. And now we try to understand how it helps us to see components in diseased states. So this is the imaging of, this is the image of a tumor cell that has been treated with our anti-cancer drug alpha-1H. Alpha-1H consists of two components or constituents, the peptide, which is shown in green, and the lipid component, the oleate, which is shown in magenta. As you can see that both the components are present in all the different parts. They are present in the cell periphery. They're very much enriched in the perinuclear region, and they are also present inside the nucleus. On the right is a merged image that gives you the co-localization of both the components, and the co-localization is shown in shades of white. So you have, wherever you have different shades of white, which is present in the periphery, in the cytoplasm, in the perinuclear region, as well as in the nucleus center. That tells you that the complex is never fully dissociated. Both the components are present in the different organelles once the cell takes off this anti-cancer molecule. On the right are shown cells that have been treated with just the individual constituents, the peptide alone in green and the lipid alone in magenta. And you can see that the uptake is minimal, which tells you that the complex is extremely crucial for the uptake, and the individual constituents are not taken up by the cells. Here is a high-resolution 3D reconstruction of the nucleus of a treated cell. And these reconstructions allow you to understand the spatial organization of the components inside the cell. And on the right, I've made the nucleus transparent, so you can actually appreciate that these constituents are inside the nucleus. This is another way to see the uptake inside. And again, this is through the reconstructions where I have taken a cross-section to see the uptake of the complex inside the cell. So those were the uptake images, but the cancer cell undergoes a huge change in morphology, as you can see, upon uptake of the anti-cancer drug. And this morphology change can be very well visualized using imaging techniques. And this is in yellow is the cytoskeleton that shows similar topology as you can see in textbook images. Now, we have used the imaging technology to not only address cancer or drugs that treat cancer, but also infected cells. And we have a pipeline of molecules from nice bacteria that provide protective immunoregulatory functions, reduce inflammation, and imaging has been used to understand the uptake of these molecules as well. Here is a cell in which in pink you see the molecules from the nice bacteria, and you can see that they are actually taken up inside the cells and how beautifully they are enriched around the nucleus. And this is used, and live cell imaging has been used to do this. This is gives you again a flavor of the power of the technology. And this is another cell in which you can see the nuclear entry of this. The nucleus is shown in green, the nuclear entry of this bacterial proteins, which are basically RNA Pol II inhibitors and reduce inflammation. So in a nutshell, I hope I have been able to convince you of the power of imaging with the state-of-the-art microscopy that we have here. And this is critical for mechanistic studies, allows us to address effects in animal models, and is essential to evaluate effects in clinical trial, which my colleague Ines would further show you. Thank you.
Any questions? Any questions?
Any questions?
There are actually some questions online, but I think maybe we will continue, Ines, with your part. What do you say?
No.
And then we can take all the questions afterwards. We're really interested in the discussion with you, so please stay on.
Yeah.
Thank you.
So Ines, please.
Yeah. So as Arima mentioned, I would like to illustrate how we use this imaging technology to look at the clinical samples. And here we show a picture of alpha-1H, so the drug, in the treated tumor tissue. So in red is the alpha-1H staining. And you can appreciate so here where the cells, so the blue nuclei are very dense, there is red staining, which means there is the drug is taken up in the tumor tissues from the treated patients. Yes, next one. But first, a small introduction of what is actually a placebo-controlled clinical trial. So in this case, patients with bladder cancer are enrolled and blindly, so nobody knows, but they're assigned to either a placebo group where they don't receive the active drug or a treatment group where they receive the alpha-1H. So everybody is blinded, so it's a double blinded study. So the patients, the doctors, even us that analyze the samples until the end of the study, where we unblind and we can compare the groups between the treatment effect between the placebo and the treatment group. So I know you've seen this slide, but I want to go through a little bit in details of the alpha-1H treatment trial. So we received two different types of samples here in the lab. First, we have the samples that are after the installation. So the patients receive either the alpha-1H, so the drug or the placebo, 6 times during a month. And we receive urine samples before they get the treatment and after they get the treatment. So we can really see and follow the response of to the treatment. And we also get the tumor response after the end of the treatment, where we get the tumor biopsy directly from the tumors that are left in the bladders of the patients. And we analyze those samples with this amazing imaging technology. So here, the first one we look at, it's the alpha-1, the drug, in the samples, the urine samples. So here I show an example of the samples before the treatment, before they receive the drug, and just after they receive the drug, so around 2 hours. And you can appreciate the red staining here is the drug, the alpha-1H, that is present in the cells that are shed in the urine of the patients. But we don't see that in the patient that received just the placebo. And we were able also to evaluate the dose response effect. So we have increased uptake of the drug with increasing dose of the alpha-1H. We also quantify this response. So with those urine, we can really count the cells one by one and look at the difference the same way. So before the alpha-1H treatment or after the treatment. And in this case, we also compare to the placebo samples. And the same way, we see a very strong dose-dependent increase in this cell shedding in the urine samples of the treated patients. Another type of analysis that we look at, it's what is called a tunnel staining. So it's shown in green here. And it looks at the cell death of the cells and the apoptosis, which is a programmed cell death, and it's a response from the body to get rid of the unwanted cells. And in this case is the cells that are, how should I say, damaged beyond repair. So the body really gets rid of those cells with this cell death response. And the same way, we see a very strong apoptosis signal in the shed cells, and this is increased with the increasing dose of the alpha-1 treatments. And amazingly, when we looked at the tumor biopsies, so those are the tumor tissues that are left after the treatment, we also see this tunnel staining, so the apoptosis, cell death signal in the leftover tumor tissue in the treated patients, but this was not seen in the placebo group. So this is really the active response in the tumor tissue to the alpha-1 drug. So the purpose of this presentation was really to present the imaging technology. So I will not go in the details of the result of the clinical trial, but I mean, we are so excited with those results that I want to reiterate that we see a very strong response in 82% of the tumors treated with the high dose of the drug. So very exciting for us to study that in details. And yeah, talking about imaging, look at this one. So this one is also, how should I say? The imaging is also used for us to look at new molecular markers of the response from those treated patients. So here is an example where we can see really the detachment of the cells from the tumor tissue and how we can analyze those images and staining with informatics tools that change the contrasts, for example, here. So there is a lot more to tell you about the drug development and the clinical trials. For example, next time we will talk about the big data analysis that we use for the infection and the cancer treatments. So we will really look forward to seeing you next time. Thank you.
Thank you, Ines. And Arunima. Yeah. So, Jakob, do we have any questions online? Okay, so I misunderstood. I thought there were questions online. Is there anyone who would like to comment or question? We're very interested in your interest in this format, which is new to us, and also we hope that this way of presenting our technology platform is interesting and also illustrates the competence and seniority of the individuals who are involved in this kind of work. I hope you really enjoyed the images from Arunima and the new insights into how cells work, and also from Ines, the fact that we can actually see things, not just in the lab, but also in the real world, in the treated patients. So we'd be happy to have your comments, by email or otherwise, and let us know if you find this exciting and worthwhile. So thank you. So there is Crester. Did you want to say something? No? Alex, thank you very much,
Thank you.
Alex, for your positive response. All right, everybody, please let us know, and we'll be back to you with this part of the series in about 1 month. There will surely be other news in the meantime. Bye-bye.
Thank you.
Bye-bye.